Welcome to the Love and Guts podcast. Martha, I keep wanting to call you Carlin. I don't know why. It's your last name, so I had to look up there. Welcome. I'm so excited to have this conversation with you.
Thanks so much for having me. I'm
And
looking
before
forward.
we get into the juicy questions, I'm going to read out your bio just in case there may be the odd few that may not know who you are. So, again,
Okay.
if I'm looking up there, guys, I'm just looking at the notes. Martha Carlin is a microbiome researcher and founder of the BioCollective, one of the first independent IRB-approved human microbiome research initiatives. Her work began after her husband was diagnosed with Parkinson's disease at 44, which led her to spend more than a decade studying microbial ecology, metabolic networks, and the biological systems behind chronic disease. She led the development of True Matrix, the first standardized human microbiome sample used to support microbiome research globally, backed by 1.2 million NIH grant. Martha also founded BioTiQuest. Is that how you say it? BioTiQuest, where she develops targeted probiotic formulations built around microbial guilds designed to restore specific biological functions. She's also a research fellow at the Australian National University and frequently speaks on microbiome science, metabolic health, and systems-based approaches to chronic disease. So there you have it. So I want to dive right in to your husband's diagnosis back in 44 of Parkinson's disease. What was the medical system's response, and what was the first thing that made you think there had to be a root cause that they weren't looking for?
Well, what's surprising to me. So the medical system, they sort of lay it out. You've got Parkinson's, and they just kind of drop it on you, and they say, there's no cure. It's a progressive neurological disease. We've got some medications that can help with symptoms, but nothing that will really stop the progress. And, here's a pill we'll see in 6 months or a year. And so they don't give you any kind of hope that there's anything that you could take action about,
Yeah.
which is and even now, 24 years later, it's still largely the same when somebody goes into a clinical neurological practice. There are many things that have happened over the last 24 years in terms of understanding these underlying factors. And there are things that you can take action to have a different trajectory, but they're not in the medical clinics.
So you just, after that clinical setting, you walked out and you're like, no, I'm not just going to accept that. Let's
Well,
just,
and that's so my background was kind of as a business turnaround expert,
yeah.
problem solver. I was also trained as an auditor coming out of college. And when you're trained as an auditor, they teach you first and foremost, never take anything at face value, examine the evidence for yourself. And
Mm.
so I really started digging into, just trying to see what the evidence was. And I was actually very fortunate. A week after John was diagnosed, I had this planned yoga retreat in Costa Rica. And
Mm.
so I went ahead and went to my retreat. And one of the people there was talking about nutrition. And they were talking about the work of Dr. Weston Price, if you're familiar with him.
Yes.
Um, and nutrition and degeneration and what had happened with the Western diet. And so I feel very fortunate that in such close proximity to what happened with John, it sort of opened up my whole world. I came back from the retreat and bought his books and started reading about it and really changed our entire diet. And one of the things I figured out very early on, so John had been drinking a soy protein shake that he was getting from a multi-level marketing company one of our neighbors was selling, and he'd been having at least one soy protein shake a day and a lot of soy milk. And that was about the time here in the States that the Roundup Ready soybeans had
Mm.
become about 97% of the market. So there was a high level of glyphosate,
Yeah.
the herbicide exposure. and of course the GMO soybeans. And so I went very deep down the rabbit hole of looking at genetically engineered food and how could that possibly be having an impact on us. And so for many years I was kind of searching for the answers to that. And then, the microbiome came along and I was like, ha, there it is.
Yeah, God, there's so much to get into there. And we're definitely going to unpack the glyphosate thing because I think that's really important for us to discuss for sure in those environmental impacts. But you've described a pivotal moment in 2014 when you came across some research linking gut bacteria to two phenotypes of Parkinson's disease. And I'm going to refer to Parkinson's disease as PD because I think I'll just be tripping up. It's such a long word. Can you walk us through what you found and why that connection felt so important?
So I had a few months earlier, I had read a book called Missing Microbes, and that sort of opened my eyes to the microbiome. And then this paper came out, Dr. Philip Schepperhans from the Helsinki University published it. And there's two types of Parkinson's, or that's sort of classically how they're separated. One is what most people think of Parkinson's, which is the tremor dominant, where you see the tremor in the hand. The other is what's called posture and gait. And that's where they're bent over more like in a penguin stance and the feet freeze. And in his paper, he showed that he could distinguish those two groups of symptomology simply by the gut bacteria. And so that for me was like, wow, okay, that is for me coming from an accounting background, I was like, this is the general ledger of the body. That's how, the record is there. And if you can separate the two phenotypes of Parkinson's, imagine what else you could do. And of course, from that was right at the sort of the wrapping up of the human microbiome project here in the States. And, there was just a huge explosion of research in not just Parkinson's, but in all these different chronic diseases. And since that paper in 2014, there have been, I want to say, about 2,000 papers published on different aspects of Parkinson's microbiome and the implications of that.
And so is this different to that body first, gut first picture versus the brain first, CNS first type?
Well, yeah, I'll say yes and no, because when they talk about body first and brain first, and that goes back to what's called Brock's hypothesis, where, well, even Parkinson's initially said that the gut was primarily involved. But, what they come up with is this body first, brain first, is a microbe is either coming through the nose and into the brain, and that pathology is presenting first, or it's coming through the mouth and into the GI tract. And then the progress there is a little bit different in the impact of the motor symptoms and the movement up through the vagus nerve into the brain. And then, you can, of course, have both body and brain first.
Oh, yeah, absolutely. And so do you think identifying those two different types is important when it comes to targeting treatment?
I think it could be down the road because coming through the nasal passage and being so close to the brain here versus what's going on in the gut, this is just my hypothesis that an intervention in this nasal area would probably need to be identified pretty quickly and taken care of, where the progress through the gut is slower.
Yeah. And so you mentioned some findings in the gut. What were they? Because, when I think about some of the signs or patterns that they might find in the gut would be like increased akkermansia or reduced short-chain fatty acids and intestinal permeability and elevated levels of hydrogen sulfide and those sorts of things. Is this the kind of things that you discovered? Like, were those the sort of things that were discovered, or was there something else?
So in our our cohort, so we did a collection over 7 years of fecal samples, not just Parkinson's, but in our Parkinson's research, we did some machine learning, and some of the things that we identified actually that haven't shown up in other publications is connection to mycobacteria. So, and I actually just put up a blog post that's a chapter out of a book I'm writing called Everything is Tuberculosis. But through the process of identifying genes involved in mycobacteria, we went through a much deeper dive of doing something called qPCR, which is looking for very specific, we probably don't need to go way down that technical
You can
rabbit
if you
hole.
want. You can if you want.
But then, and then we sent samples to another researcher who had done very specific identification of different species of mycobacteria, and actually found out of our cohort, we had 5 of the Parkinson's patients with evidence of mycobacteria, and my husband's had the highest count. It was a species called Mycobacterium lentiflavum. And so I went on kind of pushing through that and doing different types of lab research, and then got connected to someone at the University of Georgia who had developed an assay for identifying latent tuberculosis in blood, and sent John's samples to him for that. And those samples were run in triplicate 3 times, and all 3 were positive. So we went through a process of going to a tuberculosis specialty hospital, and because they were not able to grow it from his sputum, they didn't treat him for it. And so part of my hypothesis around, if you look what happens to Parkinson's, it's a very slow progressing disease. Well, mycobacteria is a very slow growing pathogen, and it was called consumption. Well, if you look at the trajectory of Parkinson's in the late stages of Parkinson's, it's very much a consumptive disease. The whole body is being consumed. Now, am I saying that mycobacteria is the cause of Parkinson's? I'm not saying that. I do believe that that is part of the ecosystem dynamic in some people. Yeah. And so that was part of what we saw. But, the other things we've seen from our research and other people's research is the elevated Akkermansia, the lower butyrate producing bacteria, damage to the gut lining, so that leaky gut, higher hydrogen sulfide production, urease producers, excuse me, methanogens. So the hydrogen sulfide and the methane and the H. pylori, which is a urease producer. Yeah. Those are all, problems in the small bowel, which, we call small intestinal bacterial overgrowth. And there's some interesting connections there, because in the last several years, there have been some fecal transplant studies in Parkinson's, and one that came from the bottom up and one that came from the top down. And the one that came from the top down was much more successful than the one that came from the bottom up, because if you're doing a fecal transplant from the bottom up, you're not getting all the way into the small bowel. So I think, as we continue to see that research progress, you'll start to see more people looking at, is the disease really up here in the small bowel? And you're not seeing a lot of that in a fecal sample.
Yeah. So, what are you seeing in a fecal sample? Because obviously we're looking at the large bowel. Are you still seeing elevated levels of hydrogen sulfide and Methanobrevibacter species being produced?
You do see that. You do see that, but you don't always see H. pylori. You won't always see H. pylori, and you won't see the mycobacteria often, because 1, it lives in the lining of the gut, and 2, its cell wall is very hard to break up lyse, which is the process of breaking up the microbes in the process for sequencing. You have to break the cell walls and get all the DNA. And
Mm.
mycobacteria is very difficult to break the cell wall. And so it often doesn't get picked up unless very specialized techniques are used in the extraction process. And so I think that is one of those things that gets missed in the evaluation of that.
Mm. And what do you think of the evaluation of PD at the moment? Because it's probably has a long way to go still.
I think it still has a long way to. In fact, I was talking to a young onset person on Sunday, and he was describing because his doctor had called to, just get some background for me, and so we were talking about it, and he was describing his first visit at the neurologist, and he said, "Well, they have you do some of these dexterity." I said, "We call it the hokey pokey."
Yeah.
Because they're like, touch your nose, tap your head, tap your foot, do so it's a very subjective evaluation. They have started now doing what's called a DAT scan to determine what area of the brain is affected. And that's interesting to me because my John was actually involved in a Department of Defense study that was a very large scale that did brain scans as a part of that study. And in that, it was over 800 people who were in this study, and they identified 15% of the people with Parkinson's they determined actually didn't have Parkinson's. They had no dopamine damage.
Mm.
And we weren't really told that when we did the study back in 2004 and 2006. But I later went back to do some digging into and research about the actual study. And at the I guess the second study point with John, he was dropped out of the study, and we were told that the study was ended, but it actually wasn't ended. Over 715 people continued on in the study for subsequent years.
Mm.
And they had this subset of people that they had identified that didn't have the dopamine loss. And so I don't really know all the background about that, but ultimately that has gone on to inform this use of the DAT scan as a diagnostic tool. But I personally think the microbiome could be a much more
Oh, yeah.
effective diagnostic tool, especially early on, because chronic constipation.
Yes, that was my next question because that's why one of the major reasons that I thought this would be a great conversation because I love treating chronic constipation.
Well, it can precede a diagnosis of Parkinson's by 10 to 15 years.
Yeah.
And often people who've been constipated that long think of it as normal, and doctors will often tell them it's normal. And it's just crazy to me.
Yes.
Um, and in our lab, when we were collecting samples, because we collected the whole sample, when they were shipped into us, my team actually came to me and said, Martha, we can tell a person has Parkinson's just from looking at their stool. It's the consistency of concrete. So
Wow.
that goes back to the battle in the gut between the good and bad bacteria and also damage to our own cells from the pore forming toxins in some of these bacteria. What happens is it spills potassium and calcium into the gut, and it just takes a tiny bit of calcium to make a concrete like colloidal mass in the stool.
Mm. So fascinating. So, why do you think that chronic constipation precedes that onset of motor symptoms by up to 20 years or more? And like, why do you think it's actually happening in the gut during those preceding years?
Well,
Do we have an
so
answer for that?
I don't know that we have a solid answer for it, but what I will say is just the fact that the waste is sitting there and you have a
Mm.
lot of endotoxin producing bacteria.
Yeah.
So produce lipopolysaccharide, and that is highly inflammatory. And so the longer it sits, the more the body is having to deal with the LPS endotoxin. And there are actually animal models of Parkinson's using endotoxin to
Mm.
cause Parkinson's like symptoms. So to me, I mean, when I found out there was an endotoxin model for Parkinson's, I'm like, okay, you make these models. Why do you not ask yourself, okay, if I can make this as a model, like what's happening in the human that's doing the same thing. It's just kind of nuts to me.
It totally is. Yeah, and look, it makes sense from a if we're looking at the large bowel and we're looking at, if someone is chronically constipated and it's sort of the perfect environment for excess methanogens to be produced and, potentially hydrogen sulfide and all of those sorts of things. So all of those little patterns that you see, as you said, in the small intestine, but potentially in the large intestine, that could be patterns that we might see in a Parkinson's picture.
Yeah.
Um,
Well, and hydrogen sulfide too is, it's a signaling molecule, but it's also a free radical scavenger.
Yeah, so tell
So,
us more about that. I want to really get into the hydrogen sulfide picture, the research around it and how it disrupts mitochondria and how that impacts PD.
so there was a group in Finland who actually came out and said this disulfovibrio is the cause of Parkinson's. And
Wow.
so I had a lot of people reach out to me and go, is that true? And I think it's, again, that whole larger ecosystem picture. So that's part of the picture.
Mm,
And when you've got oxidative stress and redox imbalance, the whole ecosystem is trying to deal with that problem. And so, a small amount of hydrogen sulfide is okay. I mean, it's
yeah,
kind of part of the system. What happens is when you tip over into a large scale production of hydrogen sulfide, and of course, hydrogen sulfide is a lethal gas. Same, the same with methane. So to me, it's all about balance in the system. And there are so many ways that you can focus on gut health and impacting change early on in the process. So
absolutely.
that to me is if they would just start to look at the microbiome early on and say,
Yes.
this is a pattern. I mean, I was looking at somebody recently who did a 90-day carnivore diet.
I was going to ask you about the carnivore because I'm not a fan, but I
Well,
for a period of time,
well,
therapeutically, potentially for some, but not a long term diet or people just popping themselves on it because they hear it's great for weight loss or, cognition or whatever it might be. The microbiome and the collateral damage to the microbiome, I just shudder. But I would love to hear your thoughts on the carnivore.
so basically what I saw was a pattern that looked very similar to a Parkinson's microbiome, which is protein fermentation instead of carbohydrate fermentation. And when you're fermenting proteins, like if you know anything about composting, you don't put protein in a compost pile. Uh, it produces ammonia, and ammonia is very toxic. And then the body's got to have to do all these things to deal with the ammonia. And so while I do think, a carnivore or a ketogenic diet can be beneficial because it takes away those carbohydrates that are feeding the pathogens in a narrow window of time, I think a long-term carnivore diet can be quite detrimental to the microbiome and the production of those short chain fatty acids.
Absolutely. And as you mentioned, just want to highlight narrow window of time.
Yeah.
And um, hopefully working with someone to guide you through when to stop, and maybe reassessing the microbiome and looking at what it looks like after you've been on a period of time on the carnivore diet. I think in a perfect world, it would look like that. But I'd love to hear what if you don't mind sharing, what sort of diet, knowing the information you did about the mycotoxins and the hydrogen sulfide and whatever was presenting in the microbiome of your partner, what sort of diet did you guys lean towards? Was it Mediterranean? Did you kind of go a bit
We
hardcore or
actually
something?
followed a lot of the dietary recommendations of the Weston Price, kind of the whole foods, real food. But there is a Dr. Terry Walls. I don't know if you're familiar with
Yes.
her, but had
MS.
reversed her MS with a nutrient dense diet that is an incredible amount of vegetables, but also, I mean, it's a paleo, so it is it is a higher fat, but she's very specific about the meats. It's a lot more lamb, some, I'm trying to think, some fish, but you're eating probably 8 cups of vegetables a day. So we did try to focus on that quite a bit in the last 4 to 5 years. We also did some work with somebody who had worked with, I think his name is Jeremy Nicholson. It's a lipid replacement diet. And that, as a part of that, one of the things we had determined was that John was actually hypoglycemic. And so trying to do more frequent feeding.
Yeah.
But when you start to have the damage to the gut lining, and it really, what I didn't really grasp all of the parts of the problem with the gut lining and the glycocalyx, which coats everything in the body, until really the last 2 years. In the last 2 years, kind of all those pieces have come together. But when you've got that damage, you're not able to take up nutrients.
Yeah, absolutely.
So part of we have a Parkinson's paper that's going to be published in the next week or two, I think. And part of the premise behind there, I was talking about the calcium and potassium spilling out. So the potassium actually stretches out the glycocalyx and stiffens it. And that's, the glycocalyx is it's kind of a molecular sieve that is sorting what nutrients get in and out. And when it gets stiff, you're not getting nutrition into the body. So John was always hungry, always hungry. And he could eat and eat and eat and not gain weight. But he wasn't absorbing nutrients. And I think that is really underappreciated. And so that, Terry Wall's diet is very nutrient dense. But the other sort of piece to that, at least here in the States, I'm not sure about Australia, but our food supply is not anywhere near as nutrient dense as it was 50 years ago. Our soil is depleted from glyphosate binding up the minerals, and just from monocropping and overgrowth of how we've done our agriculture. And so you have to eat more food to get the nutrient load, or you've got to supplement. And supplementing is problematic as well, because you end up with these kind of large swings by taking the supplements. And I think one of the things that we noticed as the Parkinson's progressed is there's a smaller and smaller window of equilibrium, of balance. And so it doesn't take much to swing you over in one direction or another, whether that's, metabolic acidosis or alkalosis, or, some other type of thing that just kind of throws the whole system off. And it's very hard to maintain equilibrium.
And so, and you've mentioned Terry Walls and fats. Were you concerned about having too much, like excess amounts of like saturated fats in that intestinal lining and hydrogen sulfide production? Or were you quite particular with those fats that you chose? Was it more olive oils? Was it more, those sorts of things? I know that could be
more olive oils, omega-3s, but those can be tricky because of oxidation. So trying to eat more fish. John did not like fish. So and then with Parkinson's, there's also quite a bit of evidence, and John had a number of tests with high mercury or high heavy metals. And so you have to be careful with the fish with that. It's like, okay, well, what can I eat? And John would joke about, this is no fun diet number 20. Like, what are we doing this week? No. But he really did. He liked the Jeremy Nicholson diet, I think, because he got to eat every 2 hours. So he
Yeah.
was eating every 2 hour. But I mean, that was really exhausting for me, trying to sort of figure out and prepare all these different, very specific foods. And again, it was a lot of vegetables and it was very
Mm.
specific fats. There's a lipid supplement that they recommend called NT Factor that's replacing the phospholipids. And I think it's a very interesting protocol, but you really need a lot of help and support
Mm.
to, execute
Implement
something
it.
like that.
Yeah.
Yeah. And a lot of people with Parkinson's live alone, or, if they're older and their spouse is older, it's a lot to try to get them to undertake something like that.
Absolutely. I was very lucky to have you, I'm sure. Someone that was so hungry for like the knowledge and to dig a bit deeper. So I'd love to hear a bit more about that, the research that you've been doing on the framework that places glycocalyx at the center of Parkinson's pathology. When's that going to be available for us to read?
Well,
And what
we got the notice today from Advances in Colloid and Interface Science that it has been accepted. So I think it will be available online probably in the next 2 or 3 weeks. They did say, so Dr. Ninham's birthday is April 8th, and they told him it would be published before his birthday. So
and I don't know whether you mentioned it, but is there a window where we can actually restore the
you
glycocalyx?
Yeah, so you can't. So this is what's so interesting. So um, Dr. Hans Vink, a researcher from the Netherlands who's here in the States now, was really the pioneer in identifying the glycocalyx in the vasculature. And he spent 30 years developing techniques to actually film the glycocalyx in blood vessels, so you
Wow.
can actually see it.
Mm.
And you can look up online Hans Vink glycocalyx, and his images are easily found now. And it looks like these fuzzy little hairs.
Mm.
And the probably the first 15 or 20 years, people just kept saying he was crazy. And now everywhere you turn, they're talking about the glycocalyx. And he has a partner. They've actually developed a product to support the glycocalyx. So the main components are hyaluronic acid, which is a fiber that only comes from animals.
Mm.
And we make hyaluronic acid. This is also, I'm going to do a little sidebar here, but in our microbiome data in our stool collection, we did a aged cohort analysis for a grant application. And one of the things that we noted as people were aging is, one of the markers was actually a drop in one of the precursors for hyaluronic acid production in the gut.
Mm.
Um, so hyaluronic acid, and then the little fuzzy hairs are made of sulfated polymers. So chondroitin sulfate, heparin sulfate, and keratin sulfate. And so, sulfur metabolism and sulfation is very important. So, back to those hydrogen sulfide guys that, they're disrupting sulfur metabolism and
Yeah.
wreaking havoc on those sulfated polymers. But seaweed, so the fucoidins of seaweed are basically sulfated polymers like that. And so there's a supplement called Revasca,
Mm.
and we're actually getting ready to do a small study in Australia in the Northern Territories in the indigenous population with a probiotic that I made for John and the Revasca, the two of those together, looking to see if they can help generally with that. And Hans actually has a device called a glycocheck that is a camera that films the blood vessels under your tongue. So if you look under your tongue, you can see there are different size blood vessels under there. You see the bigger fat ones, medium ones, and small ones. And that camera can show the damage to the microvasculature. And
Mm.
so what they have seen is, you can restore it by, essentially feeding the raw materials that help build the glycocalyx.
Mm. And so you've been looking just like beyond just diet and probiotics to environmental exposures, and we touched on it briefly, the glyphosates. So based on your work, how significant do you think glyphosate exposure and other agrochemicals are as contributors to the kind of dysbiosis we see in Parkinson's patients? And what does that mean practically for someone trying to reduce their risk, which I think is really important for the audience?
I think it's a very significant contributor. Glyphosate is actually patented as an antibiotic. And if you look at the sort of spectrum of organisms that it kills the good bacteria. So it kills those lactobacillus and bifidobacteria that are so important to, good gut health. And it leaves behind a lot of those LPS producing gram-negative bacteria. So I think it's a huge contributor, and there's lots of pushback here in the States against the people who are trying to reduce the amount of glyphosate, but it's been rising exponentially since it first came on the market. And it's not just glyphosate. There are so many chemicals in our food supply, in the herbicides, in the fungicides, in the chemicals that are used in grains that are stored to keep the from growing. When you start to dig deeper and deeper into the what's done to the food supply, and that's why I really do encourage people not to eat processed foods. If you're going to eat any kind of grains, you need to be very clear about the farmer that you're getting it from, what the process of growing those grains is. What, are they using any kind of herbicide in the drying process, because that's going to leave more residues at the end. And that's probably one of the bigger issues with most of the grains is the
Mm.
herbicide residues that are on there and the damage it does to the microbiome.
Mm. And so if people are kind of like, well, I can't eat all organic, obviously we're trying to avoid processed as much as you can. What they can probably do is start with like the clean 15 and the dirty dozen. You guys use that one too?
Yeah.
So
Yeah, and I think we may have a download still on our website where you can get the Dirty Dozen or the Clean 15, but you can look that up, and the Environmental Working Group has information on that. And
based on
um,
location too, which is I think probably important when it comes
so
to.
there are also some bacteria that can break down glyphosate.
Mm.
So Acetobacter, which is in vinegar. So I think that's probably one of the reasons why when people take apple cider vinegar, that's beneficial for them. We actually made a formula, the formula I made for John, which is our sugar shift formula. We have done research in the lab showing our Lactobacillus plantarum strain breaks down glyphosate and breaks down its secondary byproduct, which is called AMPA, which is even more toxic. And then the formula together actually breaks down about 27% of the glyphosate in 24 hours in the lab.
So
And,
what's the, I don't know. Oh, you can obviously share it because it's on your label. But why was it called the sugar shift? And what are the strains of probiotics you were using in there? We talk a lot about strain specificity here. So I'd love to hear what sort of strains you found to be impactful for this sort of.
so it's an 8 strain formula, and it's a bit like a fermented food.
Mhm.
So, we build them as teams, and so I designed it originally. So the name Sugar Shift came because I had gone to a Parkinson's conference, and they were showing some research that the sugar alcohol mannitol could stop the aggregation of the proteins in an
Mm.
animal model and actually pull the aggregates out of the brain. And I was like, okay, this is really interesting. So I came back and I bought a mannitol chemistry book, this little book. And
I
the
love the
first,
hunger for all the information. It's like, right, onto it straight away.
the first chapter of the book was about bacteria that can ferment mannitol from glucose and fructose. And I was like, oh, Well, we have way too much glucose and fructose in our diet, and I know people with Parkinson's are sugar addicts. So, can we make a team of microbes that can basically convert the sugars in the gut? And I had a friend who was fermentation chemist, and we started, kind of noodling on it and came up with this formula. And so the three of the major strains that produce mannitol from glucose and fructose are Bacillus subtilis, which is a soil-based organism, um, Lactobacillus reuteri, and I think Leuconostoc mesenteroides was the other one. And Leuconostoc is, you won't find it in most probiotics, but you will find it in almost universally in fermented foods around the world. It is the first colonizer that acidifies the environment and also makes dioxygen. So it's sort of setting up the groundwork for the whole rest of the team to come in. So your other lactobacillus. So then we have a Lactobacillus paracasei. We have our Lactobacillus plantarum that breaks down the glyphosate and AMPA. We have a Pediococcus acidilactici, which that's a
sugar shit.
That's a strain that you will often find in fermented meats.
Mm-hmm.
So it can help break down proteins, which, again, we know in Parkinson's they were having some trouble with putrefaction, so not able to break down those proteins. So you have that one. So let's see, that's for, I said Lactobacillus reuteri. And reuteri is actually one of the really popular ones today, because it's Dr. Bill Davis has kind of been the I'll say the poster person for bringing that forward. It increases the production of oxytocin. And it's a microbe that we used to have, but it's very susceptible to antibiotics. We were colonized from our mothers, and now in the Western world, a lot of people no longer have Lactobacillus reuteri. So I'm trying to think, what did I miss?
I can bring it up. I'm just pulling it up now.
I can probably bring it up. The bifidobacteria. So we have a bifidolongum and a bifidobifidum. And so those bifidobacteria are also very important.
Well to remember all those.
Yeah.
Okay, you just pull it up. So you've got the Lactobacillus reuteri PCR7. So that's the strain in the Lactobacillus plantarum LP36.
But this one, so we made it to convert these sugars, but what we found is in that whole ecosystem, it was also restoring the TCA cycle for energy production. So people were feeling better energy. It was, I think, mopping up the glyphosate. So there's a detoxification process. Some of those strains are also really effective at dealing with heavy metals. So, aluminum and mercury. So, if you have a healthy gut ecosystem, or you have a probiotic that's working like a healthy gut ecosystem, it's taking care of all those things.
Yeah.
It is helping to pull out the heavy metals. It is detoxifying the chemicals that, any sort of xenobiotics that you are exposed to. And, that's a big part of what the gut does. But also, these organisms are B vitamin producers. So there was a big paper, I think last year, it might have been the year before, on Parkinson's, and, looking at microbiome studies, and that reduction of B vitamins is a big, piece of the puzzle there. And so, you start to put back a team that can make those vitamins and convert the sugars and restore that sort of virtuous cycle instead of a vicious cycle, because there are bacteria in the gut. And I don't remember the ones off the top of my head, but one of the hydrogen sulfide producers may be one of these that run the Krebs cycle backwards, which basically is using up your energy. So you can get something called a feudal cycle, where substrates are feeding into each other, but you're not getting any energy production.
Oh, so fascinating. So I know that we wanted to talk a little bit about cardiovascular and the link between PD and cardiovascular health, if you're happy to run through that.
Yes. So I really didn't know this. And so John passed away in 20. So John got COVID at the end of 2021. So December of 2021, had a very severe case of COVID and had a lot of long-term COVID symptoms, many of which are cardiovascular,
Mm.
but weren't really, I mean, we hadn't really identified this cardiovascular component. And COVID does a lot of damage to the glycocalyx. So Parkinson's already has an unstable, sort of damaged glycocalyx. And then you have COVID, and it's damaging the vasculature. Parkinson's often has low blood pressure. And John had a pulmonary embolism in September of 2024 and passed away. And so in my mind, I'm thinking, okay, was that a fluke or not? It actually happened the day before Hurricane Helene made landfall here in the States. It was the largest barometric pressure drop in recorded history in the United States. And I remembered then, a couple of weeks later, my father had also had a heart attack that was caused by a clot. 19 years earlier, the day before Hurricane Katrina made landfall, when there was a giant drop in barometric pressure. So I started talking to Dr. Nenham, who I wrote the Parkinson's paper with, about this. And, you know, these forces that we don't see because we're swimming in air.
Yeah.
But as we started to kind of peel back the layers and look at what was going on in the glycocalyx in Parkinson's, and even experiments that John had done in the last year when we were trying to figure out electrolyte balance and stuff, as we started to look deeper into the impact on the glycocalyx and what's going on in the vascular, all of those pieces started to kind of come together. And there's a place called Parkinson's Point in Colorado that my husband was very involved in getting set up, that it's an exercise and kind of support facility. And I was there last month doing a talk, and the executive director pulled me aside and she said, Martha, in the last year, 5 of my Parkinson's patients have all died of the same thing John did. They had a pulmonary embolism. And, here in the States, each one of these things, if you're even seeing a cardiovascular, if you have Parkinson's, it's probably likely you're not seeing a cardiovascular person. They don't do blood work. Typically, neurologists don't do blood work. So there's like all these missing pieces of markers and things that
Yes.
could be identified way ahead of time that
Yeah.
nobody's looking at. So it's really kind of crazy to me.
That's
So now
strange
that
to me too.
so many people who've either had COVID or had the vaccine that have some kind of vascular inflammation in the, from the spike protein, I think it's really important for people to be hyper aware of their cardiovascular health and supporting the glycocalyx.
I think all of the, yeah, in a perfect world, you'd want to have all of the workups done, just to,
Right.
yeah, I'm sorry to hear that's happened, in
But
2020.
it was just, it's kind of crazy to me that in, 20 some odd years of going to the neurologist, I think we had blood work done once at a neurology appointment.
Wow.
And you think like, okay, endotoxin is actually something you can measure in the blood, serum endotoxin is something you can measure in the blood.
Mm. God, yeah. But even looking at the sort of the nutrient uptake and all of those sort of things that maybe you can kind of get hints on in blood work as
Right.
well. So, yeah, that's really interesting that there was only like a small percentage of time that blood work was done. So many gaps to fill
Yeah.
in this area.
And really points where you can intervene and change the trajectory. So I talk a lot to young onset people now because you get that diagnosis and it's just like this horrible bomb and you're like, oh, and but there's so many things you can do with diet and
Absolutely.
lifestyle and understanding how your body works. And then there's a whole nother piece of the puzzle that so we've been doing Voices of Parkinson's documentary, interviewing people with Parkinson's to tell their story because that story's not really getting out. And in the process of doing that, we've interviewed about 35 people. And, I would say 85 to 90% of those conversations, there was some kind of major stress event, traumatic event, you know, personal loss, business loss, something happened to a child, major divorce, all of these major stressors. And there is like unprocessed grief
Mm,
and trauma in the body. And that's a whole nother area that,
absolutely.
allopathic medicine does not look at.
Yeah.
That is just a such an important piece of the whole person and the healing process.
I think that that just extends to every condition, that sort of the emotional, spiritual, that component, mental health component is just so important. Um, I'm conscious of time. It's probably heading towards 10 p.m. for you. So I've got one more question for you, if it's okay. Just to
Sure.
bring it home. So if someone is listening today, which has received that Parkinson's diagnosis, or a family member who has, and they want to take a gut-informed root cause approach alongside conventional care, where do you actually tell them to start?
That is actually really a tough one because there aren't a lot of experts on it, but there's a couple of neurologists here in the States there's a Dr. Greg Eccles in Park City, Utah, who has just come out with kind of a platform of 5 different things, and it can be these environmental toxins, so he lays out and you could have all 5 of them or 2 of them or 3. And so he works and has had a lot of success with his patients doing much better than average. Dr. Lori Mishley, who's been an early pioneer in Parkinson's, she's a naturopath
Mm.
in either Portland or Seattle, but she sees people remotely. She also has an app called the ProPD app that has a lot of her dietary findings, and you can use that as a tool. And she has tons of online education. And then there's a Dr. Brendan Crawford, I think is in Texas, and he works a lot with autistic children, but also with people with Parkinson's. And if you have what I would say is interview a naturopath or a functional medicine doctor and get some background on what their knowledge and training is in
Yeah.
looking at the gut and success with the gut. There's also a great podcast here in the States called The Perfect Stool. The woman's name is Lindsay Parsons, and she does a lot of work. She's probably one of the more knowledgeable people, when I listen to people talk about the gut and looking at the gut and what they've done. And so you can learn a lot from her, but she also does, like, she'll do, a 3 session kind of look at your gut and give you some ideas. And so you can find her online as well.
So what I'm hearing as well is that, like what you mentioned before, that it's not, hopefully people just aren't walking away feeling helpless. They've been given a diagnosis or a family member, and you just go, okay, well, we just kind of have to enjoy the rest of our lives because we just don't know what's going to happen. There is so much you can do, with just gather the information, like you said, go to the professionals that know what they're doing when it comes to the gut microbiome, get your blood work done, all of that, as we see that that's important to do as well. So there's so much that you can do, and it's people hopefully feel like there's a little bit of hope, you know.
Yes, I think there's a lot of hope, and um, certainly go enjoy your life. And
Yes.
that's um, one of the biggies because John used to say sort of the 80-20 rule is that 80% of the people would take that diagnosis and just go sit in the chair and wait to die.
Yeah.
And the other 20% of the people were out trying to figure out, what can I do about this? How can I take action? Oh, we didn't even talk about exercise. There's biking programs and boxing and different things that, use both sides of the body. But, your life's not over. You can have lots of adventure. John and I had tons of adventures after he was diagnosed with Parkinson's. And so, your life just isn't over, and there's a lot you can do.
Amazing. So what's next for you, and where can people find you? Obviously, that's going to be in the show notes, but what's next for you? Of
Well, I actually have a book coming out.
course you do.
I hope
Papers
in
and books.
May. Uh, I finished the first draft of it today. It's gone to the editor,
Oh,
and
amazing.
that's really the journey with John and all of the things that I have learned along the way that hopefully can be helpful for people. I also have a small book called In Search of the Perfect Poop that just, or no, sorry, My Search for the Perfect Poop, that's available now on Amazon. And
Amazing.
it really just talks about how important looking at your stool and knowing, am I going regularly? So you can find that. I write general health and gut health blog content on our BioTiquest blog. That's BioTiquest.com. And I have a Parkinson's blog called Martha's Quest. So you can find me there as well.
And I'll make sure I pop all that in the show notes. So I do enjoy your blogs, by the way. I think they're great source of information. So
Thank you.
thank you so much for joining us so late in the evening for you, especially after you've just handed in the first draft of your book. So I'm even more impressed now. Thank you so much for joining us today, Martha.
Thanks for having me, Linda.
You're very welcome. And guys, if you've enjoyed this conversation or you know someone in your life that needs to hear it, please make sure you share it with them. And while you're at it, please subscribe to Love and Guts so that others can fill up on the valuable content that Martha has presented to us today.